A pleasant day to everyone. We are Group 1 and we are going to present about neurons and synapses.
Everyone, this is Ann Abarico, one of the reporters from Physiological Psychology B1, and I welcome you to Chapter 2, The Cells of the Nervous System. Today, we will be tackling nerve cells and nerve impulses. Ready? Let's go! People talk about growing into adulthood and becoming independent, but in fact, almost no human life is truly independent. How often you hunt your own meat and cook it on a fire you made from scratch? Do you grow your own vegetables? Could you build your own house with tools you made yourself? Of all the activities necessary for your survival, which ones, if any, could you do completely in your own other than breathe? People can do an enormous amount together, but very little by themselves. The cells of nervous system are like that too. Together, they accomplish amazing things, but one cell by itself is helpless. We begin our study of the nervous system by examining single cells. When you think yourself as an individual, you don't think of your mental experience as being composed of pieces, but it is. Your experiences depends on the activity of a huge number of separate but interconnected cells. Let's examine and understand the cells of the nervous system by this topic outline. The two kinds of nervous system is called neurons and glia, wherein neurons are capable of receiving information and transmit it to other cells, whereas glia serve many functions that are difficult to summarize, and we shall defer that discussion until later in this module. The adult human brain contains approximately 86 billion neurons in average. We now take it for granted that brain is composed of individual cells, but the idea was in doubt as recently as the early 1900s. Until then, the best microscopic views revealed little detail about the brain. In the late 1800s, Santiago Ramon y Cajal, a pioneer of neuroscience, used newly developed staining techniques to show that a small gap separates the tip of neuron's fiber from the surface of the body, consists of individual cell. Cajal, along with Charles Sherrington, are the two scientists of the late 1800s and early 1900s, and are widely recognized as the main founders in neuroscience. Cajal is a Spanish investigator who wanted to become an artist, and by his father, he studied medicine and combined the two fields. He became an outstanding anatomical researcher and illustrator. His detailed drawing of nervous system are still considered definitive today. In 1800s, microscopy revealed few details about the nervous system. Camillo Golgi, an Italian investigator, found a way to stain nerve cells with silver salts. Golgi's method, used by Cajal in an infant brains in which the cells are smaller and therefore easier to examine on a single side. Cajal's research demonstrated that nerve cells remain separate instead of merging into one another. The structures of an animal cell. Neurons have much in common with the rest of the body cell. Membrane or plasma membrane separates the inside cell from the outside environment. Most chemicals cannot cross the membrane, but protein channels in the membrane permit a controlled flow of water, oxygen, sodium, potassium, calcium, chloride, and other important chemicals. All animals have nucleus, a structure that contains the chromosomes. Mitochondria or mitochondrion is the structure that performs metabolic activities, providing the energy that the cell uses for all the activities. People whose mitochondria are less active than normal are predisposed to depression and pains. Ribosomes is capable of synthesizing new protein molecules. Some float freely within the cell. Others are attached to endoplasmic reticulum. Proteins provide building materials for the cell and facilitate chemical relations. Endoplasmic reticulum a network of thin tubes that transport newly synthesized proteins to other locations. The structures of a neuron. The most distinctive feature of neurons is their shape, which varies enormously from one neuron to another. All neurons include soma or cell body, and most also have dendrites, an axon, and presynaptic terminals. The tiniest neurons lack axons, and some lack well-defined dendrites, contrasting motor neuron and sensory neuron. Motor neuron, with its soma in the spinal cord, receives excitation through its dendrites and conducts impulses along its axon to a muscle, whereas a sensory neuron is specialized at one end to be highly sensitive to a particular type of stimulation, such as light, sound, or touch. Conducts touch information from the skin to the spinal cord. See figure below. Other parts of neuron includes dendrites, or branching fibers that get narrower near their ends. Greek root word meaning tree. A dendrite branches like a tree. Many dendrites contains dendritic spines. This is a short outgrowths that increase the surface area available for synapses. The cell body or the soma, Greek for body, plural, somata. This contains the nucleus, ribosomes, and mitochondria. Metabolic works occur here. In many neurons, cell body is like the dendrites, covered with synapses on its surface. The axon is a thin fiber of a constant diameter. The term axon comes from the Greek word meaning axis. The axon conveys an impulse toward other neurons, an organ, or a muscle. Many vertebrate axons are covered with an insulating material called myelin sheath, with an interruptions known as nodes of Ranvier. Invertebrates' axons do not have myelin sheath. Presynaptic terminal, also known as an end bulb or button, a swelling of each ending branches. At that point, the axon releases chemicals that cross through the junction between the neuron and the other cell. Other terms associated with neurons are afferent, efferent, and intrinsic. Afferent axon brings information into a structure. Efferent axon carries information away from a structure. Every sensory neuron is an afferent to the rest of the nervous system, and every motor neuron is an efferent from the nervous system. You can remember that efferent starts with A and an exit. Afferent starts with A as an admit. Intrinsic neuron or interneuron if a cell's dendrites and axon are entirely contained within a single structure. Glia or neuroglia, components of nervous system which performs many functions. The term glia derived from a Greek word meaning glue. Glia were like glue that held neurons together. Although that term is obsolete, the term remains. Glia outnumber neurons in the cerebral cortex, but neurons outnumber glia in several other body areas, especially cerebral. The brain has several types of glia. These include the star-shaped astrocytes, wrap around the synapses of functionally related axons. By surrounding a connection between neuron, an astrocyte shields it from chemicals circulating in the surround. Astrocytes are therefore important for generating rhythms, such as your rhythm in breathing. Astrocytes dilate blood vessels to bring more nutrients into the brain area that heightened activity. In some brain areas, astrocytes also respond to hormones and thereby influence neurons. In short, astrocytes are active partners for neurons in many ways. Microglia. Tiny cells act as part of immune system, removing viruses and fungi from the brain. They proliferate after brain damage, removing oligodendrocytes in the brain and spinal cord, and Schwann cells in the periphery of the body build the myelin sheaths that surround and insulate certain vertebrate axons. They also supply an axon with nutrients necessary for proper functioning. Radial glia guide the migration of neurons and their axons and dendrites during embryonic development. When embryological development finishes, most radial glia differentiate into neurons, and a smaller number differentiate into astrocytes and oligodendrocytes.
Hello everyone, I am Rhea Aganap, and in this slide I am going to tackle all about the blood-brain barrier. Like any other organ, the brain needs to receive nutrients from the blood. The mechanism that excludes most chemicals from the vertebrate brain is known as the blood-brain barrier. But before we examine how it works, let's consider why we need it. When a virus invades a cell, mechanisms within the cell extrude virus particles through the membrane so that the immune system can find them. The purpose of the blood-brain barrier is to protect against circulating toxins and pathogens that could cause brain infections while at the same time allowing vital nutrients to reach the brain. The vertebrate brain does not replace damaged neurons. To minimize the risk of irreparable brain damage, the body lines the brain's blood vessels with tightly packed cells that helps the brain to stay away from harmful chemicals like viruses and bacteria. The spirochete, a flexible spirally twisted bacterium that causes syphilis, also penetrates the blood-brain barrier, producing long-lasting and potentially fatal consequences. Lastly, the microglia are more effective against several other viruses that enter the brain because it helps to fight viruses without killing the neurons. The blood-brain barrier depends on the endothelial cells that form the walls of the capillaries. In the brain, endothelial cells are packed much tighter together, blocking the passage of most molecules. The blood-brain barrier blocks all molecules except those that cross cell membranes by means of lipid solubility like oxygen and carbon dioxide, and those that are allowed in by specific transport system like sugar and amino acid. For these chemicals to cross the blood-brain barrier, the brain needs special mechanisms not found in the rest of the body. How fast a drug takes effect depends largely on how readily it dissolves in fats and therefore crosses the blood-brain barrier. The advantages and disadvantages of blood-brain barrier is that it keeps out viruses and also keeps out most of the nutrients. However, the barrier poses a difficulty for treating brain cancers because nearly all the drugs used for chemotherapy fail to cross the blood-brain barrier. Here's an illustration to understand more about how the blood-brain barrier works. Why do neurons depend so heavily on glucose? Most cells use a variety of carbohydrates and fats for nutrition, but vertebrate neurons depend almost entirely on glucose or sugar. Although neurons requires glucose shortage is rarely a problem except during starvation. To use glucose, the body needs vitamin B1 and thiamine. People who has a chronic alcoholism leads to death of their neurons and a condition called Korsakoff syndrome, if you heard about that. This is a memory disorder caused by severe deficiency of thiamine and vitamin B1. We have here the neurons. One of my members already discussed this in the previous slides, but let me give you a brief summary about neurons. Neurons receive information and convey it to other cells. Santiago Ramon y Caja used newly discovered staining techniques to establish that the neuron system is composed of separate cells now known as the neurons. And because of the blood-brain barrier, many molecules cannot enter the brain. The barrier protects the nervous system from the viruses and many dangerous chemicals. So that's it for this slide. I hope you learned something from me today. Thank you!
Hi, Day. Today, we'll be talking about nerve impulse. An axon conveys information from the touch receptor in your hands or feet towards spinal cord and brain. It is an electrical conduction if its axon can transfer informations at velocity approaching the speed of light. But because our body is not made up of copper, instead it is made up of water and carbon compounds, so its impulse strength would decay rapidly as it travels. So to avoid these kinds of action, our axon regenerates an impulse at each point. Think of a long line of person holding hands, then the first person squeeze the next person, and so on. Because each person regenerates the impulse, so even if it travels along the line, it is not getting weak. Axon transmit information at only moderate speed, from less than 1 meter per second up to 100 meter per second. It is the reason why a touch on your shoulder reaches sooner than a touch on your foot, although you cannot identify it. And in your vision, according to Stanford 1987, axons from more distant parts of your retina transmit impulses slightly faster than those closer to your brain. The properties of impulse conduction in an axon are amazingly well adapted to your needs.
The resting potential of a neuron. Messages in a neuron develops from disturbances of the resting potential. Let us understand the resting potential. All parts of a neuron are covered by a membrane about 8 nanometers thick. It is about 1/10,000 the width of an average human hair. The membrane is composed of 2 layers, which is free to float relative to each other, of phospholipid molecules, which is containing chains of fatty acids and a phosphate group. Embedded among the phospholipids are cylindrical protein molecules through which certain chemicals can pass. Let us see figure 1.12. The electrical gradient, or also known as polarization, is the difference of the electrical charges of the cell between its inside and outside. The electrical protein inside the membrane is slightly negative compared to the outside. It is because of the charge of the protein inside the cell which is negative. This difference in voltage is called the resting potential. And the researchers uses a very thin microelectrodes to measure the resting potential of the body. Let us see figure 1.13, which shows methods for recording activity of a neuron. Forces acting on sodium and potassium ions. In selective permeability, some chemicals pass through it more freely than others do. The membrane would depolarize if the charged ions can flow freely across the membrane, eliminating the negative potential inside. Oxygen, carbon dioxide, urea, and water are one of the biologically important ions that can cross freely through channels that are always open. Sodium, potassium, calcium chloride can cross through membrane channels that are sometimes open or closed. It is shown in figure 1.14. A sodium-potassium pump is a protein complex repeatedly transports 3 sodium ions out of the cell while drawing 2 potassium ions into it. It is an active transport that requires energy. As a result, sodium ions are more than 10 times concentrated outside the membrane, and potassium ions are more concentrated inside than outside. Sodium-potassium pump is effective only because of the selective permeability of the membrane. If the membrane is at rest, the sodium-potassium pump draws the potassium ions into the cell. Forces acting on sodium and potassium ions. When the neuron is at rest, 2 forces acting on sodium. Both tends to push it into the cell. First to be considered is the electrical gradient. Sodium is positively charged and the inside of the cell is negatively charged, and remember, opposite electrical charges attract, so the electrical gradient tends to pull sodium into the cell. Secondly, consider the concentration gradient, the difference in distribution of ions across the membrane. Sodium ions are more concentrated outside the cell, and potassium is more concentrated inside. Potassium is subject to competing forces. Potassium is positively charged and the inside of the cell is negatively charged, so the electrical gradient tends to pull potassium in. But because potassium is more concentrated inside the cell than outside, so the concentration gradient tends to drive it out. For an analogy, imagine a number of women inside a room. Men can enter the room or leave through a narrow door. They are attracted to the woman, but when the men get too crowded, some of them leave. The concentration gradient counteracts the attraction. When the membrane is at rest, the concentration gradient tends to drive potassium ions out of the cell, and the electrical gradient draws them into the cell. Why resting potential? The body invests so much energy to operate sodium-potassium pump, which maintains the resting potential. It invests too much energy because it prepares the neuron to respond rapidly. Compare the resting potential of a neuron to a poised bow and arrow. An archer who pulls the bow in advance is ready to fire at the appropriate moment. The neuron uses the same strategy. The resting potential remains stable until the neuron is stimulated. Ordinarily, stimulation of the neuron takes place at synapses. In the laboratory, it is also possible to stimulate a neuron by inserting an electrode into it and applying current. The action potential are the messages sent by the axon. The hyperpolarization is an exaggeration of the usual negative charge within a cell to a more negative level than usual. Depolarization is a decrease in the amount of negative charge within the cell. Graph 1. When an axon membrane is at rest, the recording shows a negative potential inside the axon. If we now use a different electrode to apply a negative charge, we can further increase the negative charge inside the neuron. The change is called hyperpolarization, which means increased polarization. When the stimulation ends, the charge returns to its original resting level. Graph 2. To depolarize the neuron is to decrease its polarization towards zero. The graph shows a small depolarization current applied on the neuron. Graph 3 shows a graph that has a slightly stronger depolarization current. The potential rises slightly higher but again returns to the resting level as soon as the stimulation ceases. Graph 4 shows a still stronger current. Stimulation beyond the threshold of exciting produces a massive depolarization of the membrane. When the potential reaches the threshold, the membrane opens its sodium channels and lets sodium ions flow into the cell. The potential shoots up far beyond the strength that is the stimulus provided. Any subthreshold stimulation produces a small response that quickly decays. Any stimulation beyond the threshold regarding of the how far beyond produces a big response like the one shown, known as the action potential. The peak of the action potential shows as 130 MV in this illustration varies from one axon to another. The all or none law. The amplitude and velocity of an action potential are independent of the intensity of the stimulus, provided that it reaches the threshold. By analogy, imagine flushing a toilet. You have to make a press of the least a certain strength, the threshold. But pressing harder does not make the toilet flush faster or more vigorously. Similarly, when you flick the switch to turn on your lights in your room, flicking the switch harder would not make the lights brighter. Amplitude, velocity, and shape of action potential are consistent over time for a given axon, but they vary from neuron to another. According to all or none law, the size and shape of the action potential are independent of the intensity of the stimulus that initiated it. That is, every depolarization beyond the threshold of excitation produces an action potential of about the same amplitude and velocity for a given axon. The nervous system uses both kinds of coding. For example, a taste axon shows one rhythm of responses for sweet taste and a different rhythm for bitter taste. And all or none law does not apply to dendrites because they do not have an action potential. Molecular basis of the action potential. To make a complex one simple, take note of these three principles. First, at the start, sodium ions are outside and potassium ions are inside the neurons. Second, When the membrane is depolarized, sodium and potassium channels in the membrane open. Third, at the peak of the action potential, the sodium channel close. A neuron membrane contains cylindrical proteins. A sodium channel is a protein that allows sodium to cross. And potassium channel, it is a protein that allows potassium to cross. The voltage-gated channels are the axon channel regulating sodium and potassium. The movement of sodium and potassium ion during an action potential. Figure 1.16 summarizes the key movements of ions during an action potential. At the end of this process, the membrane has returned to its resting potential, but the inside of the neuron has slightly more sodium ions and slightly fewer potassium ions than before. Eventually, the sodium-potassium pump restores the original distribution of ions, but that process takes time. After an unusual rapid series of action potentials, the pump cannot keep up with the action, and sodium accumulates within the axon. Excessive buildup of sodium can be toxic to a cell. Excessive stimulation occurs only under abnormal conditions, however, such as during a stroke or after the usage of certain drugs. Action potential requires the flow of sodium and potassium. Local anesthetic drugs, such as Novocaine and Xlocaine, attach the sodium channels of the membrane, preventing sodium ions from entering. When a dentist administers Novocaine before drilling into one of your teeth, your receptors are screaming pain, 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 but the axons cannot transmit the message to your brain and so you don't feel it.
Good day. This is the continuation of Module 1.2, the nerve impulse. Propagation of the action potential. Describes the transmission of an action potential down an axon. The propagation of an animal species is the production of offspring. In a sense, the action potential gives birth to a new action potential at each point along the axon. According to Lawrence and Miller, 2010, an action potential always starts in an axon and propagates without loss from start to finish. However, at its start, it backpropagates into the cell body and dendrites. Backpropagation is important. When an action potential backpropagates into a dendrite, the dendrite becomes more susceptible to the structural changes responsible for learning. When an area of the axon membrane reaches its threshold of excitation, sodium channels and potassium channels open. Opening of potassium channels produces little effect, but sodium channels let sodium ions rush into the axon. The membrane is now depolarized. Positive charge flows down the axon and opens voltage-gated sodium channels at the next point. At the peak of the action potential, the sodium gate snaps shut because voltage-gated potassium channels remain open. Potassium ions flow out of the axon, returning the membrane toward its original depolarization. A few milliseconds later, the voltage-dependent potassium channels close. Myelin sheath and saltatory conduction. In the thinnest axons, action potentials travel at a velocity of less than 1 meter per second. Increasing the diameter brings conduction velocity up to about 10 meters per second. At that speed, an impulse along an axon between a giraffe's spinal cord and its foot takes about half a second. To increase the speed still more, vertebrate axons evolved a special mechanism: sheaths of myelin, an insulating material composed of fats and protein. Myelinated axons, found only in vertebrates, are covered with layers of fats and proteins. The myelin sheath is interrupted periodically by short sections of axon, called nodes of Ranvier, each one about 1 micrometer wide. The action potential cannot regenerate along the membrane between nodes because sodium channels are virtually absent between nodes. After an action potential occurs at a node, sodium ions enter the axon and diffuse, pushing a chain of positive charge along the axon to the next node, where they regenerate the action potential. Saltatory conduction, jumping of action potentials from node to node, comes from Latin word saltare, meaning to jump. Saltatory conduction conserves energy. Instead of admitting sodium ions at every point along the axon and then having to pump them out via the sodium-potassium pump, a myelinated axon admits sodium only at its nodes. If the nodes were closer, the action potential would travel more slowly. If they were much farther apart, they could travel faster. Refractory period. A period that happens after the sodium gates snap shut after the peak of the action potential. It is when the cell resists the production of further action potential. It has two types. Absolute refractory period is when the membrane cannot reproduce another action potential, regardless of the stimulation. It lasts for about 1 millisecond. On the other hand, relative refractory period requires a stronger than usual stimulus to initiate an action potential. It lasts for about 2 to 4 milliseconds. The refractory period depends on two facts. 1. Sodium channels are closed, and 2. Potassium is flowing out of the cell at a faster rate than usual. Local neurons are small neurons that has no axon. They exchange information with only their closest neighbors. They do not follow the all or none law. Graded potential is a membrane potential that varies in magnitude in proportion to the intensity of the stimulus.
If you had to communicate with someone without sight or sound, what would you do? Chances are your first choice would be a touch code or a system of electrical impulses. You might not even think of passing chemicals back and forth. Chemicals are, however, the main way your neurons communicate. They communicate by transmitting chemicals at specialized junction called synapses. Hi, my name is Angelo Ariola, and my topic is to discuss the concept of synapse as well as its properties. In the late 1800s, Ramon E. Cajal anatomically demonstrated a narrow gap separating one neuron from another. In 1906, Charles Scott Sherrington physiologically demonstrated that communication between one neuron and the next differ from communication along a single axon. He inferred a specialized gap between neurons and introduced the term synapse to describe it. Cajal and Sherrington are regarded as the great pioneers of modern neuroscience, and their nearly simultaneous discoveries supported each other. Sherrington's discovery was an amazing feat of scientific reasoning, as he used behavioral observation to infer the major properties of synapses. That's half a century before researchers had the technology to measure those properties directly. Sherrington found that the velocity of a conduction through a reflex arc was slower than the velocity of an action potential along an axon. Therefore, some delay must occur at the junction between and one neurons and the next. This happens in a junction called synapse. Sherrington also studied reflexes, like automatic muscular responses to stimuli. The circuit from a sensory neuron to muscle response is called reflex arc. If one neuron is separated from another, as Cajal had demonstrated, a reflex must require communication between neurons, and therefore measurement of reflexes might reveal some of the special properties of the communication. Sherrington observed several properties of reflexes that suggest special processes at the junction between neurons. Number one, reflexes are slower than conduction along an axon. 2. Several weak stimuli presented at nearby places or times produce a stronger reflex than one stimulus alone does. Number 3, when one set of muscles become excited, a different set becomes relaxed. Now for the first property of synapse. 1, like what was mentioned, speed of a reflex and delayed transmission at the synapse. Sherrington measured the total distance that the impulse traveled from skin receptor to spinal cord to muscle and calculated the speed at which the impulse traveled to produce the response. He found that the speed of conduction through the reflex are varied but was never more than 15 meters per second. In contrast, previous research had measured action potential velocities along sensory or motor nerves at about 40 meters per second. Sherrington then concluded that some process must be slowing conduction through the reflex, and he inferred that the delay occurs where one neuron communicates with another. This idea is critical as it established existence of synapse. Sherrington in fact introduced the term synapse. For the second property of synapse, it's the temporal summation, which means summation over time. Sherrington found that repeated stimuli within brief time have a cumulative effect. He coined this phenomenon as temporal summation. The neuron that delivers transmission is the presynaptic neuron, while the one that received it is called the postsynaptic neuron. Sherrington proposed that although the subthreshold excitation in the postsynaptic neuron decays over time, it can combine with the second excitation that follows it quickly. With a rapid succession of pinches, each adds its effect to what remained from the previous one, until the combination exceeds the threshold of the postsynaptic neuron, producing an action potential. Decades later, Sherrington's former student, John Eccles, attached microelectrodes to stimulate axons of presynaptic neurons while he recorded from the postsynaptic neuron. Note that this partial depolarization is graded potential, unlike actions potential, which are always depolarization graded potential, may be either depolarization, which means the excitatory, or the hyperpolarization, or which means inhibitory. A graded depolarization is known as excitatory postsynaptic potential. It results from a flow of sodium ions into the neuron. If an EPSP does not cause the cell to reach the threshold, the depolarization decays quickly. The third property of synapse is the spatial summation. Sherrington also found that synapse have the property of spatial summation, which means summation over space. Synaptic inputs from separate locations combine their effects on a neuron. Sherrington concluded that pinching 2 points activated separate sensory neuron whose axon converge onto one neuron into the spinal cord. Excitation from either sensory axon excited the spinal neuron, but not enough to reach the threshold. A combination of excitation exceed the threshold, and produce an action potential. Spatial summation is critical to brain functioning. In most cases, sensory input, a single synapse produces only a weak defect. However, if a neuron receives many incoming axons which synchronize input, spatial summation excites the neuron enough to activate it. Temporal summation and spatial summation ordinarily occur together. That is, a neuron might receive input from several axons in close succession. Integrating this input provides complexity. The next property of synapse is the spatial summation. Sherrington also found that synapses have the property of spatial summation, which means summation over space. Synaptic inputs from separate locations combine their effects on a neuron. Spatial summation is critical to brain functioning. In most cases, sensory input at a single synapse produces only a weak effect. However, if a neuron receives many incoming axons with synchronized input, spatial summation excites the neuron enough to activate it. Now, temporal summation and spatial summation ordinarily occur together. That is, a neuron might receive input from several axons in close succession. Integrating this input provides complexity. Another property of synapse is the inhibitory postsynaptic potential. It is a kind of synaptic potential that makes a postsynaptic neuron less likely to generate an action potential. Inhibitory synapses influence signals in the brain with high precision. In our brain, information is passed from one cell to the next via a trillion of synapses. However, optimal data flow is not just about the transfer of information, its targeted inhibition is also a key factor. Basically, an inhibitory postsynaptic potential is a kind of synaptic potential that makes postsynaptic neuron less likely to generate an action. Inhibitory synapses is a temporary hyperpolarization of membrane. It's an active brake that suppresses excitation, which leads to the absence of excitation. It occurs when synaptic input selectively open the gates for potassium ion, which carries positive charges, or for chloride ions to enter the cell, which carries negative charges.
Now that we already learned what FPSP, IPSP, and action potential are, let us know the relationship of these three potentials. The work of Sherrington opened the way to the exploration of nervous system's wiring diagram. Considering the presented diagram, when neuron 1 excites neuron 3, it also excites neuron 2, which inhibits neuron 3. As you can see, excitation reaches the dendrite before inhibition, and the excitatory message reaches neuron 3 faster because there is only one synapse instead of two. As a result, excitation bursts in neuron 3, which quickly slows or stops. Synapses vary enormously in the duration of effects. Some synapses produce fast with brief effects, while others produce slow but have long-lasting effects. Various inputs interact in ways that are not quite additive. The effect of two synapses at the same time can be more than double the effect of either one, or less than double. To further understand how the wiring diagram in the nervous system controls the result, let us consider the presented diagram. The axon from either cell A or cell B stimulates cell X with +1 unit. If the threshold of cell X is +1, then cell X responds to A or B. If the threshold is +2, then cell X responds to A and B. Thus, it depends on whether the threshold for cell X is 1 or 2. It responds to A or B, or it responds to A and B. Neurons have a spontaneous firing rate, and it refers to its rate of producing action potentials even when it is not stimulated. In such cases, the epiSPs on a neuron compete with the IPSPs. The balance between the two increases or decreases the neuron's frequency of action potentials. While epiSPs increase the frequency of action potentials above the spontaneous rate, IPSPs decrease the number of action potentials below the spontaneous rate. The neuron as decision maker. Synapses are the ones who decide whether or not to send messages that will be transmitted through an axon from one place to another. epiSPs and IPSPs that arrive at a neuron at the same time compete with one another, resulting in a complex but non-algebraic summation of their effects. Hence, their summation can be considered as a decision since it decides whether or not the postsynaptic cell fires an action potential. The discovery of chemical transmission at synapses. Charles Sherrington has accurately distinguished numerous synapse properties. However, he missed one important thing, and that is, he concluded that synaptic transmission relies on electrical process and not on chemical process. German physiologist Otto Lowy was the first to convincingly demonstrate that communication across a synapse occurs via chemical means. In his experiment, which he used two frogs' hearts, he demonstrated chemical transmission by repeatedly stimulating frog's vagus nerve. That makes the frog's heart rate decrease. He then collected fluid from around that heart, applied it to the second frog's heart, and found that the second heart also decreased its rate of beating. In a related experiment, Lowy stimulated the accelerator nerve to the first frog's heart, increasing the heart rate, and just like what he did previously, he collected fluid from that heart and transferred it to the second frog's heart, making its heart rate increase. Lewis' interpretation of these results was that there was some substance released by the nerves he stimulated, and the fact that the substance could then be transferred to the second frog's heart made him confident that the substance he collected was a chemical. Hence, Louis concluded that the nerves send messages by releasing chemicals. In the 1950s, researchers established that chemical transmission predominates throughout the nervous system, and the great majority of synapses rely on chemical processes, which are much faster and more versatile than Sherrington or anyone else of his era would have guessed.
The sequence of a synapse. Sequence of a synapse. First, the neuron synthesizes chemicals that serve as neurotransmitters. It synthesizes the smaller neurotransmitters in the axon terminal. Second, action potentials travel down the axon. At the presynaptic terminal, an action potential enables calcium to enter the cell. Calcium releases neurotransmitters from the terminals and into the synaptic cleft, the space between the presynaptic and postsynaptic neurons. The third step, the released molecules diffused across the narrow cleft attach to receptors and alter the activity of the postsynaptic neuron. Mechanisms vary for altering that activity. The fourth step, the neurotransmitter molecules separate from their receptors. The fifth, the neurotransmitter molecules may be taken back into the presynaptic neuron for recycling, or they may diffuse away. And the last part, some postsynaptic cells send reversed messages to control the further release of neurotransmitter by presynaptic cells. Neurotransmitters. A neuron that releases chemicals that affect another neuron. 100 or so chemicals are known or suspected to be neurotransmitters, as shown in Table 2.1, which is the next slide. Example, nitric acid, which has chemical formula NO,
It is a gas released by many small local neurons. Nitric oxide, NO with nitrous oxide, N2O, known as laughing gas. Nitric oxide is poisonous in large quantities and difficult to make in a laboratory. Yet, many neurons contain an enzyme that enables them to make it efficiently. Many neurons release nitric oxide when they are stimulated. In addition to influencing other neurons, nitric oxide dilates the nearby blood vessels, thereby increasing blood flow to that brain area. More samples of neurotransmitters. First is amino acids, which consist glutamate, GABA, glycine, aspartate, and maybe others. Second is a modified amino acid, which only consists acetylcholine. Third is monoamines, also modified from amino acids. It also consists idolamines, serotonin catecholamines, dopamine, norepinephrine, and epinephrine. Fourth example is neuropeptides, chains of amino acid, which consist endorphin substance P, neuropeptide Y, and many others. Fifth is the purines, which consist ATP, adenosine, and maybe others. Last is gases, which consist nitric oxide and maybe others. Ctenophores, possibly representative of the earliest, most primitive animals, apparently have only one neurotransmitter, which is the glutamate. Most of the rest of the animal kingdom has all or nearly all of the same transmitters that humans have. Catecholamines, the relationship among epinephrine, norepinephrine, and the dopamine compounds. They contain a catechol group and an amine group as shown here in the next figure. Each pathway in figure 2.13 begins with substances found in the diet. Acetylcholine, for example, is synthesized from choline, which is abundant in milk, eggs, and peanuts. The amino acids phenylalanine and tyrosine, present in proteins, are precursors of dopamine, norepinephrine, and epinephrine. People with phenylketonuria lack the enzyme that converts phenylalanine to tyrosine. They can get tyrosine from their diet, but they need to minimize intake of phenylalanine because excessive phenylalanine would accumulate and damage the brain. Tryptophan, the precursor to serotonin, crosses the blood-brain barrier by a special transport system that it shares with other large amino acids. Your serotonin levels rise after you eat foods richer in tryptophan, such as soy, and fall after something low in tryptophan, such as maize. However, tryptophan has to compete with other more abundant large amino acids, such as phenylalanine, that share the same transport system. So increasing intake of tryptophan is not the best way to increase serotonin. Vesicles, where presynaptic terminal stores high concentrations of neurotransmitter molecules. The presynaptic terminal also maintains such neurotransmitter outside of vesicles. Neurons that release serotonin, dopamine, or norepinephrine contain an enzyme MAO, monoamine oxidase, that breaks down these transmitters into inactive chemicals, thereby preventing the transmitters to accumulate to harmful levels. The first antidepressant drugs that psychiatrists discovered were MAO inhibitors. By blocking MAO, they increase the brain supply of serotonin, dopamine, and norepinephrine. However, MAO inhibitors also have other effects, and exactly how they help relieve depression is still not certain. Exocytosis, bursts of release of neurotransmitter from the presynaptic neuron, an action potential that often fails to release any transmitter, and even when it does, the amount varies. An action potential itself does not release the neurotransmitter. Rather, depolarization opens voltage-dependent calcium gates in the presynaptic terminal within 1 or 2 milliseconds after calcium enters the terminal. After it's released from the presynaptic cell, the neurotransmitter diffuses across the synaptic cleft to the postsynaptic membrane, where it attaches to a receptor. The neurotransmitter takes no more than 0.01 millisecond to diffuse across the cleft, which is only 20 to 30 nanometer wide,
Remember, Sherrington did not believe chemical processes could be fast enough to account for the activity at synapses. He did not imagine such narrow gap through which chemicals could diffuse so quickly.
Good morning. Today, I will tackle about the activating receptors to electrical synapses. Sherrington's concept of the synapse was simple. Input produces excitation or inhibition, in other words, an on-off system. First, we have to clarify that the words excitatory and inhibitory means. Excitation is the process by which a neuron's membrane potential or voltage increases. If excitation is sufficient, a neuron will produce an action potential. Inhibition is a process by which a neuron membrane potential or voltage decreases. If a neuron is already firing, then if it receives enough inhibition, it will stop firing. So the statement is some neurons are excitatory, meaning they will fire, and some inhibitory, meaning they won't, is not quite right. All neurons, whether excitatory or inhibitory, can fire, but only if they receive adequate excitation. If an inhibitory neuron fires, it can reduce the voltage of other neurons, whether they are excitatory or inhibitory. Excitation is the accelerator for all neurons. Inhibition is the brake for all neurons. When echoes recorded from individual cells, he happened to choose cells that produce only brief EPSPs and IPSPs. Again, just on, off. An EPSP is received when an excitatory presynaptic cell connected to the dendrites fires on the action potential. Inhibitory post-synaptic potentials, or IPSP, is a temporary hyperpolarization of postsynaptic membrane caused by the flow of negative charge ion into a postsynaptic cells. Researchers discovered more and more neurotransmitter and wondered why does the nervous system use so many chemicals if they all produce the same type of message. Eventually, they found that the messages are more complicated and more varied. When the neurotransmitter attaches to its receptors, the receptors may open a channel exerting an ionotropic effect, or it may produce a slower but longer effect, a metabotropic effect. The ionotropic effect. Neurotransmitters exert ionotropic effects corresponding to the brief on-off effect that Sherrington and Eccles studied. An ionotropic effect can be applied to the effect of a transmitter substance or hormone on its target. The transmitter or hormone activates or deactivates ionotropic receptors, ligand gate ions channel. The effect can be either positive or negative, especially a depolarization of hyperpolarization respectively. For example, imagine a paper bag that is twisted shut at the top. If it untwisted it, the opening grows larger so that something can go into or come out of the bag. An ionotropic receptor is like that. When the neurotransmitter binds to an ionotropic receptor, it twists the receptors just enough to open its central channel, which has shape that lets a particular type of ion pass through. In contrast to the sodium and potassium channel along an axon, which are voltage gated, the channel controlled by a neurotransmitters are transmitter gated or ligand gated channel. Ligand gated ion channel or LGICs are one type of ionotropic receptor or channel link receptor. They are a group of transmembrane ion channel that open or close in response to the binding of a chemical messenger or ligand, such as neurotransmitter. A ligand is a chemical that binds to something. That is, when the neurotransmitter attached, it opens a channel. A eutrophic effect begins quickly, sometimes when less than a millisecond after transmitter attaches. Most of the brain's excitatory iotropic synapses use the neurotransmitter glutamate. In neuroscience, glutamate refers to the anion of glutamic acid in its role as a neurotransmitter, a chemical that nerve cells use to send signal to other cells. It is by a wide margin the most abundant excitatory neurotransmitter in the vertebrate's nervous system. In fact, glutamate is the most abundant neurotransmitter in the nervous system. Most of the inhibitory ionotropic synapses use the neurotransmitter or GABA, gamma aminobutyric acid, which open chloride gates, enabling chloride ions with their negative charge to cross the membrane into the cell more rapidly than usual. Glycine is another common inhibitory transmitter, found mostly in the spinal cord. Acetylcholine, another transmitter at many ionotropic synapses. As shown in the picture, the letter A, the acetylcholine receptors, a cross section of the receptor at rest, as viewed from the synaptic cliff. The membrane surrounds it. The letter B, a similar view after acetylcholine has attached to the side of the receptor, opening the central channel wide enough for sodium to pass through. Metabotropic effect and second messenger system. At other receptors, neurotransmitters exert metabotropic effect by initiating a sequence of metabolic reaction that starts slowly but lasts longer than ionotropic effect. Metabotropic effect emerges 30 minutes or more after the release of the transmitter. Typically, it lasts up to a few seconds, sometimes longer, whereas most ionotropic effect depends on other glutamate or GABA. Metabotropic synapses use many neurotransmitters including dopamine, norepinephrine, and serotonin, and sometimes glutamate and GABA2. Example: imagine a large room. You are outside the room holding a stick that goes through a hole in the wall and attaches to the hinge of a cage. If you shake the stick, you open the cage and release the angry dog. The dog runs around, waking up all the rabbits in the room, which then scurry around, causing all kinds of further action. A metabotropic receptors acts a little like that. When a neurotransmitter attaches to a metabotropic receptors, it bends the receptor's protein that goes through a membranes of the cell. The other side of the receptor is attached to a G protein. That is the protein coupled to guanosine triphosphate or GTP, an energy storing molecules. Bending the receptor's protein detach the G protein, which is then free to take its energy elsewhere in the cell. As in the picture, the result of the G protein is increased concentrated of a second messenger such as cyclic adenosine monophosphate or cyclic AMP. Inside the cell, just as the first messenger, the neurotransmitter carries information to the postsynaptic cell. The second messenger communicates to areas within the cell. It may open or close ion channel in the membrane or activate portion of a chromosomes. Note, an ionotropic synapse has effect localized to one point on the membrane, whereas a metabotropic synapse by the way of its second messenger, influences activity in much or all of the cell and over a longer of time. Ionotropic and metabotropic synapses contribute to different aspects of behavior. For example, for vision and hearing, the brain needs rapid, up-to-date information the kind of ionotropic synapse bring. Metabotropic synapses are also important for many aspects of arousal, attention, pleasure, and emotion. Again, functions that arise more slowly and last longer than a visual or auditory stimulus. Neuropeptides. Researchers often refers to the neuropeptide as neuromodulators because they have properties that set them apart from other transmitters. Whereas the neurons synthesize most other neurotransmitters in the presynaptic terminal, it synthesizes neuropeptides in the cell body and then slowly transports them to other parts of the cell. Whereas other neurotransmitters are released at the axon terminal, the neuropeptides are released mainly by dendrites and also by the cell body and by the sides of the axon. A single action potential can release a neurotransmitter, but neuropeptides release requires repeated stimulation. However, after a few dendrites release a neuropeptides, the release chemical primes other nearby dendrites, including those on other cells, to release the same neuropeptides also. Furthermore, unlike other transmitters that are released immediately adjacent to their receptors, neuropeptides diffuse widely, slowly affecting many neurons in their region of the brain. In that way, they resemble hormones because many of them exert their effects by alternating gene activity. Their effects often last 20 minutes or more. Neuropeptides are most important for hunger, fears, and other long-term changes in behavior and experience. The table summarizes difference between neurotransmitter and neuropeptide. Variation in receptors. The brain has a variety of receptors types for each neurotransmitter. Receptors for a given transmitter differ in their chemical structure, responses to drugs, and roles in behavior. Because of this variation in properties, it is possible to devise drugs with specialized effect on behavior. For example, the serotonin receptor types 3 mediates nausea and the drugs ondansetron that blocks these receptors help cancer patients undergo treatment without nausea. A given receptor can have different effects for different people or even in different parts of one person's brain because of differences in the hundreds of proteins associated with the synapse. The synapse is a complicated place where dozens of proteins tether the presynaptic neuron to the postsynaptic neuron and guide neurotransmitter molecules to their receptors. Genetic variation in synaptic proteins have been linked to variation in anxiety, sleep, and other aspects of behavior. Drugs that act by binding to receptors. A drug that chemically resembles a neurotransmitter can bind to its receptor. Many hallucinogenic drugs, that is, drugs that distort perception, such as lysergic acid, diethylamide, or LSD, chemically resembles serotonin. They attach to serotonin type 2A receptors and provide stimulation at inappropriate times or for longer than usual duration. LSD increases the connection among brain areas that ordinarily do not communicate much with one another. A possible explanation for the hallucinogenic effect is that the increased spontaneous communication within the brain dominates over the input coming from the sense of organ. Nicotine, a compound present in tobacco, stimulates a family of acetylcholine receptors, conveniently known as nicotinic receptors, because nicotinic receptors are abundant on neurons that release dopamine. Nicotine increases dopamine release. Because dopamine release is associated with reward, nicotine stimulation is rewarding also. Opiate drugs are derived from or chemically similar to those derived from the opium poppy. Family opiates include morphine, heroin, and methadone. People use morphine and other opiates for centuries without knowing how the drugs affected the brain. The researchers found that opiates attach to specific receptors in the brain. It was a safe guess that vertebrates had not involved such perception just to enable us to become drug addicts. Soon, investigators found that the brain produced its own neuropeptides, now known as endorphins, a contraction of endogenous morphines that binds to the same receptors as endorphins. This discovery was important because it indicated that opiates relieve pain by acting on the receptors in the brain as well as in the skin. This finding also paved the way for the discovery of other neuropeptides that regulates emotions and motivations. Inactivation and reuptake of neurotransmitter. A neurotransmitter does not linger on the postsynaptic membrane, where it might continue exciting or inhibiting the receptors. Various neurotransmitters are inactivated in different ways. The neuropeptides, however, are not inactivated. They simply diffuse away because resynthesizing these large molecules takes time and neurons can temporarily exhaust its supply. After acetylcholine activates a receptor, the enzyme acetylcholinesterase breaks it into two fragments, acetate and choline. The choline diffuses back to the presynaptic neuron, which takes it up and reconnects it with acetate already in the cell to form acetylcholine again. Although this recycling process is highly efficient, it takes time and the presynaptic neuron does not reabsorb every molecule it releases. A sufficiently rapid series of action potential at any synapse depletes the neurotransmitter faster than the presynaptic cell, replenished it, thus slowing or interrupting transmission. Serotonin and catecholamine or dopamine, norepinephrine, and epinephrine do not break down into inactive fragments at the postsynaptic membrane. They simply detach from the receptor at the point the next step varies. The presynaptic neuron takes up much or most of the released neurotransmitter molecules intact and reuses them. This process, called reuptake, occurs through special membrane proteins called transporters. The activity of transporters varies among the individual and from one brain area to another. Any transmitter molecules that the transporters do not take will instead break down by an enzyme called catechol-emit transferase. The breakdown produce, wash away, and eventually show up in the blood and urine. Stimulant drugs, including amphetamine and cocaine, inhibit the transporters for dopamine, serotonin, and norepinephrine, thus decreasing reuptake and prolonging the effect of the neurotransmitters. Most antidepressant drugs also block reuptake of one or more neurotransmitters but more weakly than amphetamine and cocaine do. When a stimulant drug increases the accumulation of dopamine in the synaptic cleft, Cohn breaks down the excess dopamine faster than the presynaptic cell can replace it. A few hours after taking a stimulant drug, a user has a deficit of dopamine and enters a withdrawal state, marked by reduced energy, reduced motivation, and mild depression. Methylphenidate, Ritalin, another stimulant drug, is often prescribed for people with attention deficit hyperactivity disorder. Methylphenidate and cocaine blocks the reuptake of dopamine in the same way at the same brain receptors. The difference between the drug related to dose and time course, cocaine users typically sniff it or inject it to produce a rapid rush or of effect on the brain. People taking methylphenidate pills experience a gradual increase in the drug's concentration over an hour or two, followed by a slow decline. Therefore, methylphenidate does not produce the sudden rush of excitement that cocaine does. However, everyone who injects methylphenidate experiences effects similar to cocaine, including a risk of addiction. Negative feedback from postsynaptic cell. Suppose someone sends you an email message that then worry that you might not have received it. Send it again and again to prevent cluttering your inbox. You might add a system that provides an automatic answer. Yes, I got your message. Don't send it again. A couple of mechanisms in the nervous system serve that function. First, many presynaptic terminals have receptors sensitive to the same transmitter they release. These receptors are known as autoreceptors, receptors that respond to the release transmitter by inhibiting further synthesis and release, that is, they provide negative feedback. Second, some postsynaptic neurons respond to stimulation by releasing chemicals that travel back to the presynaptic terminal to inhibit further release of transmitter. Nitric oxide is one such transmitter. Certain cells in the retina emit hydrogen ions, that is protons, to inhibit further transmission. Two other reserved transmitters are anandamide, from the Sanskrit word anana meaning bless, and 2AG. Cannabinoids, the active chemical in marijuana, binds to anandamide or 2AG receptors on presynaptic neurons, indicating the cell got your message, stop sending it. The presynaptic cell, unaware that it hadn't sent any message at all, stopped sending. In this way, the chemical in marijuana decreased both excitatory and inhibitory messages from neurons that release glutamate or GABA and other transmitter. In various causes, the result can be either brief or more long-lasting suppression of release. The behavioral results vary but usually include decreased anxiety. This picture summarizes some of the ways in which drug affects dopamine synapses, including effects on synthesis, release, action, and postsynaptic receptors, reuptake, and breakdown. This table summarizes effects of some common drugs. Electrical synapses. At the start of this module, you learned that Serrington was wrong to assume that synapses convey message electrically. Well, he wasn't completely wrong. A few special purpose synapses do operate electrically because electrical transmission is faster than even the fastest chemical transmission. Electrical synapses have involved in cases where exact synchrony between two cells is important. For example, Some of the cells that control your rhythmic breathing are synchronized by electrical synapses. It's important to inhale on the left side of the same time as on the right side. Also, many animal species have electrical synapses in their system responsible for coordinating rapid escape movement. At an electrical synapse, the membrane of one neuron comes into direct contact with the membrane of other. This contact is called a gap junction. These pores are large enough for sodium and other ions to pass readily, and unlike the other membrane channels we have considered. These pores remain open constantly, therefore, whenever one of the neurons is depolarized, sodium ions from the cell can pass immediately into the other neurons and depolarize it too. Fairly large pores of the membrane in one neuron line up precisely with similar pores in the membrane of the other cell. As a result, the two neurons act as if they were a single neuron. Again, we see the great variety of synapses in the nervous system. And that's all. Thank you for listening.
The topic that we are going to learn is about the hormones. Hormones are chemical substances that affect the activity of another part of the body. Hormonal influences resemble synaptic transmission in many ways, including the fact that many chemicals serve both as hormones and as neurotransmitters, while the nervous system uses neurotransmitters as its chemical signals. The endocrine system uses hormones. In this slide, we'll learn the differences between neurotransmitters and hormones. A hormone is a chemical secreted by cells in one part of the body and conveyed by the blood to influence other cells, while a neurotransmitter is like a telephone signal. It conveys a message from the sender to the intended receiver. Figure 2.20 shows the major endocrine hormone-producing glands. Many glands make up the endocrine system. The hypothalamus, pituitary gland, and pineal gland are in your brain. The thyroid and parathyroid glands are in your neck. The thymus is between your lungs, the adrenals are on top of your kidneys, and pancreas is behind your stomach. Ovaries in women. These organs make estrogen and progesterone. Testes in men. The testes make testosterone. There are two types of hormones, which are protein hormones and peptide hormones, composed of short chains of amino acids, whereas protein hormones are longer polypeptides. Both types are synthesized like other proteins. DNA is transcribed into mRNA, which is translated into an amino acid chain. In Figure 2.21, it shows the location of the hypothalamus and pituitary gland in the human brain. It is located at the base of the brain near the pituitary gland. While it's very small, the hypothalamus plays a crucial role in many important functions, including releasing hormones. Pituitary gland has two parts, the anterior pituitary and posterior pituitary. The anterior pituitary receives signaling molecules from the hypothalamus and in response synthesizes and secretes 7 hormones. The posterior pituitary does not produce any hormones of its own. Instead, it stores and secretes 2 hormones made in the hypothalamus. Figure 2.23 is about the negative feedback in the control of thyroid hormones. The hypothalamus secretes a releasing hormone that stimulates the anterior pituitary to release TSH, which stimulates the thyroid gland to release its hormones. Those hormones, in turn, act on the hypothalamus to decrease its secretion of releasing hormone. In closing, will be about neurotransmitters and behavior. In the century plus in Sherrington, we have come a long way in our understanding of synapses. We no longer think of synapses as simple on or off messages. Nerve cells communicates message by secreting neurotransmitters. Neurotransmitters can excite or inhibit neurons, nerve cells. Each neurotransmitters can directly or indirectly influence neurons in a specific portion of the brain, thereby affecting behavior.
Once again, this is Group 2. Thank you for listening. Wear your mask, wash your hands, and keep your distance from others. God bless.